welcome to a talk which is going to be, I think there are quite a few people here who actually know bioinformatics, so you won't like this one. But after a certain age, you can do things that you want rather than doing the things that you have to do. So I'll give you the talk that I want to give. My name is Khalid Sayood. I'm at the Department of Electrical and Computer Engineering at the University of Nebraska. And my lab is called the Occult Information Lab. The occult stands not for magic, but for hidden, because that's what we try to do, is look for information in data. A few things to start with. So I was just telling Professor Sankur that I got up at 2 this morning because of jet lag, and that probably doesn't mean that I'll fall asleep. Some of you might. But what I do is I tend to kind of wander off topic. And if that happens, I apologize in advance. The talk is going to be wandering anyway, so you might not notice the difference. But so if you have questions, please ask. Otherwise, it's going to be a very short talk. Or you can look at it differently too. If you don't ask questions, it's going to be a very short talk, whichever perspective you want to keep. Most of your English is probably better than my Turkish, but if it isn't, ask me the questions in Turkish. Anlayacak kadar Türkçem var, but not enough to give a technical talk. So, and finally, I am a very disorganized person, and one of the things that they tell you to do is, as your first slide, put down the organization. I don't have an organization, so this is my outline. So, you know, and it's redundant, you know, if you think about it. We've already begun, so that first line is useless. The middle is going to be the rest of the time, and the end is when I stop talking. Okay, that's it. Okay, so here we go. My roots are in signal processing, communications, information theory, and data compression. I started out doing adaptive equalizers for my master's, worked on vector quantization, communication stuff for my dissertation, and continued working either in the subfield of information theory or even the subfield of that, which is source coding or data compression. Okay, and that's pretty much why I'm in bioinformatics. So, sometimes it moves, sometimes it doesn't. So data compression is basically the art or science of finding compact representations of information in data. That is what you do when you do data compression. You look for where the information is, and you try to find a way of representing it that doesn't take up that much space. Because most of the time, you're not after data, you're after the information. Right? So as long as you can model the information, you can throw away the rest and you get compression. Okay, that's in essence all that data compression is. And it is mostly an art. The way you do this is, and I've done this for a long time now, you take a look at the data, and I make my students stare at their data, because once you stare at something long enough, you start seeing some kind of a structure in the data. And once you have a structure, you can model it, you can figure out how to represent that model, and then when you have to write the paper, then you go back and say, okay, mathematically this is how it all developed. You try to make it sound coherent. But first you get the algorithm, then you figure out how it's supposed to come about. So the art isn't actually figuring out the compression algorithm. The science is figuring out how to satisfy the reviewers. Okay. With information, I started out in speech compression, continued with image compression, then when video started coming in, did video compression. And when DNA sequences started appearing in the 90s, I mean, that's like, crack to an addict. There is a huge amount of data in DNA. There was no way I wasn't going to do work with DNA. So that's when I started working with DNA. sequences. I don't really do a hell of a lot of proteins. I do a little bit, not a whole lot. So you know, if you think about it, it seems like the whole bioinformatics field is set for communications. You have a DNA sequence, in that you have the messages, the genes, or you can think of the genes as the transmitter, and then you generate the messenger RNAs, and then and that can be your message, or you can look at the protein as your message, or the protein as the user. There are all kinds of ways you can set it up, right? Or you can take a look at the actual sequence, and you can think of this being a coded representation of information. Or you can think of, you know, if you're thinking of alternative splicing sites, you can think of the whole gene as being signal plus noise, with the things that you're going to splice out as the noise and the things that are going to remain as the signal. Right? So there are all kinds of ways in which this could be, you know, you can see analogies to communications. And if you, the original definition of bioinformatics, the first time the word was used, the definition that was given was that it was the study of informatic processes in biotic systems. Okay, so everything looks great from my point of view. We're looking at a communication system. We're looking at transmission of information. So, you know, I can go look at where the information is in the data, I can model it, and I can be happy the rest of my life. Things didn't quite work out that way. This is a definition from a 1999 set of slides from Mark Gerstein. And here is what bioinformatics actually is today. Conceptualizing biology in terms of molecules, that's fine. Then applying informatics techniques, and there's the air quote around informatics. You know, you can see that you're kind of moving away from information. To understand and organize the information associated with these molecules on a large scale. So bioinformatics became, how do I organize information in databases? Something that I'm sure some of you get very excited about, and I find tremendously boring. So, you know, life was bad for me for a long time. But, you know, here's more. What is bioinformatics? I stole these slides from one of my former students. He's actually a graduate of this school. If you look at it, you know, there are three parts to bioinformatics. It's the management and analysis information system. There's nothing about how to model information, how to extract information. It's just, let me analyze this data as if it was information. Let me manage the data, because I have a lot of it. Let me find matches to it. That's kind of what bioinformatics is right now. This is a workflow for a transcriptomics project. You know, you start out with mapping. You have the Burrows-Wheeler transform. You use Bowtie or BWA or SOAP2. You go through, if you have unmapped reads, you use split-seq or top hat or SOAP, some version of it. And then you go through summarization, you apply the various tools as you go along, and in the end, you have some biological insight, right? You go through, you figure out what the reads correspond to, you come up with counts of what the expression patterns are, you figure out which genes are being differentially expressed, you figure out which KEGG pathways these correspond to, and now you say, okay, these are the pathways that are active in the cancer cells, and these are the pathways that are active in the non-cancer cells. Okay, that's the standard approach. And I'm not saying we don't do that. So I asked one of my students, we're doing a transcriptomics thing for a different group, a virology group. And so I asked him to send me what he was doing. And he sent me this. He said, I use FastQC, then I use Termomatic, then I use Bowtie 2, and then I use Top Hat 2, and then I use CuffDiff. Right? Everything are tools. These are all tools. All large programs, all programs with a lot of different parameters. Most of these parameters you leave as default. You didn't even try to figure out what they are. Those that you don't leave as default, if you change them, you get slightly different results. And because there are so many of them, the cascade effect is by changing the parameters, you can get significantly different results. Right? So here's some more acronyms. I'll just leave it aside. And this is what I got. This is a school, and they're advertising their bioinformatics program. If you look at it, there's bioinformatics, there's Python, R, Linux, methods, scripts, hypothesis design, databases, project, visualization. There is the word information in there, but that's about the only word that I like. Most of the other stuff I don't. So what's wrong with that? I mean, why not do this? One thing is we don't really understand, you know, applying all of these programs one after the other, you don't really know what's going on. You know what the program output is. And the program was written by somebody with certain assumptions that are inherent in what those program outputs are. Unless you sat down and figured out that, I don't know, bow tie and top hat and cufflink and all the other things that are coming out of, what's his name? I forget his name. Saltzman. His lab are all based on Burrows-Wheeler transform. And what does the Burrows-Wheeler transform do? How does it function? And how is it used in all of these? I mean, you know, when you talk about FM index, what exactly is that in terms of, again, of the Burrows-Wheeler transform? But most people who use these programs really don't have an idea what's under that program. They know that if you use them, you'll get certain results. And you take those results as given because it's just too complicated, right? And there's I am putting it down, but it is useful. We have gotten a lot of good stuff done over the last 20 years or so because of these very large programs, because they have systematized the way we analyze data. There's a lot of good stuff in there, but there's bad stuff too. And the bad stuff is, and this you probably can't read. So here's the problem. Now, if you give me a null hypothesis, and the null hypothesis is you give me a coin, and you say, this is a fair coin. And I take that coin, and I toss it 10 times, and I get 10 heads in a row. I have good reason to reject the null hypothesis, right? I can say, I'm going to reject the null hypothesis with a high level of significance. I can calculate out the probabilities of that happening under the null hypothesis, the coin is fair, and then I can say I can reject this at this level of significance. Right? But another thing I could do is I could flip the coin 10 times, look at it, and flip it again 10 times, and keep doing that until I get 10 heads in a row. Then I can report that I flipped the coin 10 times, I got 10 heads in a row, and therefore I'm rejecting the null hypothesis. Nothing that I've said there is actually inaccurate. Nothing there is wrong. It's not the complete, you know, description of what happened, but nothing there is wrong. And because you have all of these programs one after the other, because you have this workflow in which there are all these parameters that can be adjusted, you can run that experiment many, many, many times until you get the right result with a p-value of 0.05. I think it's called p-hacking. So, or, you know, that's being malicious, that's doing this on purpose. Or you could do it really totally innocently, because you're sitting there and you're running the program and the results come out and you know what the results should be. You have in your head what you think the right answer is. So you go about and you say, yeah, you know that doesn't sound quite right. I wonder if I use this program wrong. I wonder if I change this parameter, something that would fix the problem. And totally innocently, you can go and you can totally mess up the whole idea of significance. Okay, so that's a problem. And another way of looking at it is, this basically, I'll move the data around until I get the right answer. Right? So there are all kinds of good things in the way we do bioinformatics nowadays, but there are dangers in the way we do bioinformatics nowadays. Yeah. And what I'm saying is that there is another perspective as well, and that's, of course, my perspective. Right? So is there hope for me? Of course. I'm the eternal optimist. So here's the thing. In 1975, that was a digital camera. I know because I used one. I have in my lab one of the pieces of equipment that I have not been able to throw away from olden times is a set of optical discs which record 50 seconds of color video. We used it for video compression experiments, or, just obtaining images, all kinds of stuff back in the day. And I have a really hard time throwing it away. And one of the reasons that I paid, or basically the grant paid, $78,000 for those 2 optical discs, which store 50 seconds of video. Right? And this was late 80s. Today, we're in this, actually we're a little bit further than that. If you have a MinION sequencer, it's a small sequencer, you can get long sequences from it, but we're still at the very beginning. We're at the stage where we're going to be inundated with lots and lots and lots more data. The answer from a lot of bioinformatics people is, well, that's a reason for making more and more efficient databases. No. That's the reason for maybe taking a look at how to do things differently, not discarding what we have right now. I think what we have right now has some very good things in there, but to also add a different point of view. And of course, I'm talking, so that's my point of view. Okay, so here's the thing. Oh, I love this one because, whenever something happens, they say, well, things are getting so, there's Moore's law. Computing will be fast enough. Data storage will be, dense enough. No problem. The guy who's hanging there doesn't probably believe that. So I'm not entirely sure that technology is going to save us. So what I'm going to do is give you a communication theory perspective. Remember that we have been dealing with messages, with signals, with large amounts of signals, and with the design of systems that depend on understanding those signals for a very, very long time. Okay? And so we've developed some approaches. And what I'm saying is not that these should replace what already exists. I'm saying that these are things that maybe you should consider as adjunct to what you already have. Okay? So I crossed out the we and I said, what's I? Because that's the way I think about it. So for me, information exists in the form of a stochastic process. Okay? You've got all kinds of realizations. You have, if I talk about DNA, any particular DNA sequence that I get is actually the realization of a stochastic process. And the stochastic process works under some rules, and those are the rules that we're after when we try and design communication systems. Right? So how do we handle stochastic processes? Well, a number of different ways. We try to look at the signal, the thing that we have to deal with, in a number of different ways. We have to have different ways of looking at the signal itself. In bioinformatics, mostly that means different ways of visualization. That's not what I mean. I mean different ways of looking at that sequence. Again, I'm going to be principally talking about DNA sequences here. I'm not going to be talking about proteins, or even if I talk about proteins, it's going to be primary sequences. So that's going to be my entire focus here. But if you think about it, maybe you say, how else do I look at it? You've got a sequence, and it's a sequence. You can put it in a FASTA format. You can generate it in a FASTQ format. You can, but in the end, it's a sequence. I can look at quality values maybe, but that's about it. What else do you do? There are many, many different ways of looking at it, at a particular signal. In communications, we do that by looking at that same signal using different basis sets. Right? If I use a complex exponential basis set, I get the Fourier transform. If I use different kinds of square waves, then I get a discrete Walsh-Hadamard transform. You know, I can take a signal and I can look at it in many different ways. And by looking at it in many different ways, I can see what the structure is in the data. Okay? I can look at the correlation structure. How are things related to each other? And I can try and model the data. Okay. What does that give us? Well, first of all, notice that all of these require averaging of some sort. So you're going to be giving up something, right? Remember that. You're going to give up something. But all of these result in the discovery of underlying structure in the data. By averaging through, doing that intelligently, you can discover what's the structure of the data. Okay. And also, if you get a model of some sort, the parameters of the model are much less than the dimensionality of the signal itself. So the model lives in a much, much lower dimensional space than the signal does. And that means you get this enormous amount of dimensionality reduction. Okay, so here's realizations of a stochastic process. So I said the word hello into the microphone a number of times. And these are all me saying hello. And they're all, and I didn't try to vary my sound or something. These are natural variations. They're all realizations of a stochastic process. They all contain the exact same information from our point of view. They're all somebody saying hello, but they all look different. Okay. If it's the information that I'm after, the way the signal looks is not that important. It's the information that I'm after, right? So I can look at it, I can take a look at it in different ways, like I said. I mean, you can look at it in terms of a frequency profile, and I screwed up, and I got the reflection there as well. But if you've got some kind of a frequency profile, this is the magnitude frequency spectrum. And you can see there's a lot of similarity here. There are differences, but there's a lot of similarity. Okay, maybe you can make use of that. You can look at the statistical profile. Right? So now, even though the original words were very different, this is the autocorrelation. They look very similar. Right? Or you can come up with a model. So this is a different word. It's me again. I'm always my model. So this is the word test. And what I've done is I have the frequency spectrum, and on top of that, I have the spectrum for different models. Okay, so the top left one is a third order model. The next one is a fifth order, an 11th order, and 21st order model. And you notice that as we go higher and higher, by the 21st order model, that model captures the main characteristics of the spectrum really well. Now, maybe that's not what we're after. We're after the small variations, in which case this is not a good idea. But if we're after the general shape of the spectrum, then that model contains 21 coefficients as opposed to the 4,000 frequency values that I have in the original data. So I have this huge amount of reduction in the amount of data that I have to work with. Okay, so your data is increasing daily, but the information is not necessarily increasing that fast. And so if you can figure out what is the underlying structure in your data, you don't have to deal with this information, they call it information explosion, it's actually a data explosion. Okay? So that's the main pitch that I have for you. So, but how do we do this with DNA sequences? Because we have sequences, we don't have numbers. How do we take the Fourier transform? How do we model it? Well, there are a number of different ways to do it. And I'm going to pick one, but here's quite a few of the others. So I can assign a value, a numerical value. I can say A is 0, C is 1, G is 2, and T is 3. And suddenly, instead of a sequence, I have a waveform. So my DNA sequence is now a waveform. Okay, there's an argument against that by the way I pick which number to attach to which base. I'm introducing a bias, valid criticism. But here's an alternative way of looking at it, a way that is very amenable to a lot of signal processing techniques that we already know. Okay. We can assign a direction to each of the bases. Okay, and then think of a sequence as a random walk. And people did that back again in the 90s or so. And you can look at the entire sequence as just a random walk of some sort, and then you can find the properties of that random walk and see if those properties match up with some biological properties. Okay. This is a very common way of doing it, is you assign a binary string, 4 binary strings for the sequence. So each binary string is an indicator function. The first binary string in this case just tells you where the A's are. The second binary string tells you where the C's are, G's, and T's. Right? So I've got 4 different binary strings. And because they're now strings of numbers, I can operate on them. I can use different algorithms on them. And this one was a 2015 paper, so it's pretty recent, where they used a wavelet transform to figure out the coding regions in a gene. So we have this I forget what the gene was, but this some kind of standard gene that everybody works with, which has a lot of coding and non-coding regions overlapping. And so in order to figure out where the exons and where the introns are, what you can do is you can use a wavelet transform, and wherever you have peaks there, and you have to put a threshold of some sort in there, you can figure out where the coding regions are and where the exons are and where the introns are. So, you know, you can do good things. You can do, this is now a very common thing to do, is we can take a sequence, and instead of just representing the sequence, we can look at the frequency of occurrence of, in this case I've used 2, but nobody uses 2, k-mers. We can take 3 bases, 4 bases, 5 bases, 8 bases, and then we can say how many times does this particular combination occur. And we end up, in this case, with a vector that's 16 long, because I've taken 2 bases at a time. And this vector is now a k-mer vector. And using k-mers, you can do a lot of things that you'd have to use quite heavy computations to do. So k-mers are getting to be very popular nowadays. And this is my favorite. So this is the one I'm going to focus on. So this function, forget the k for the moment. This is the average mutual information. I have 2 random variables, x and y. In communications, these are generally the input and the output of a channel. So x is the input, y is the output. The average mutual information tells you how much information is contained in random variable x about the random variable y. You okay? So I'm looking at, if I'm looking at transmission over a channel, I put in a message, which is some realization of the random variable x. I get out something from the channel, which is some realization of the random variable y. I would like at the receiver to know as much as possible about what was transmitted. So what I want to do is maximize the amount of information that gets transmitted over the channel. Well, this thing tells you how much information there is in one random variable about the other random variable. Okay, so to compute it, you need the joint probability of the 2 random variables, and you need the marginal probability. So basically, you need the joint probability, right? If x and y are totally independent, they have nothing to do with each other, then the joint probability is just a product of the marginals. So p of xy is just going to be p of x times p of y. So the numerator is going to look exactly like the denominator in that log. And because they look the same, they get canceled out, and you get log of 1, and you have a mutual information of 0. If x and y are independent, then there is no information in X about Y or in Y about X. Okay, so this is the quantity that I'm going to be using. This is my favorite quantity, so this is the quantity I'll be using to talk about sequences. It doesn't mean that this is the best way to do things. It's just the one that I like. Okay. Now, like I said, you know, you grow old enough, you can do what you like, more or less. I didn't want to wear a tie. He made me do it. Okay. Nobody has any questions? You're going to fall asleep, you know. Okay, see, here's where the K comes in. So I have a DNA sequence in this case. This could be any primary sequence. It could be a sequence of amino acids. And the K is how far the bases are from each other, right? So when I look at I1, or in this case, say I6, then what I'm trying to figure out is what is the information in G, in that first G, about that T 6 bases away. Okay, so I can compute I1, which will tell me what is the information in one base about its neighbor. I can compute I2, which will tell me what is the information contained in this base about the neighbor of the neighbor. I can compute I3, which will tell me what is the information in this base about its, you know, 3 down. You get the idea, right? So you keep going. And in this way, you build up a profile of the DNA sequence. Okay. This is an information profile, but it's just a profile of the DNA sequence. Okay. I'm going to be using this profile a lot, so are you guys comfortable with that? More or less? Yeah, just generally. Okay. So if I do this for the human genome, this is the, and I call it the AMI profile, the average mutual information profile. This is for chromosome 1. And like I said, this was around 2000 that we came up with this. The average mutual information had been used before in the context of bioinformatics, but it had been used to understand secondary structure and a few other things. But nobody had to come up with, you know, try to figure out a profile. So we came up with the idea, we tried to come up with a profile, and we got this result. And this was my first PhD student in the bioinformatics area, Mark Bauer. And what we expected was a decaying exponential. We expected a more or less smooth decaying exponential. We didn't get that. We got these little peaks and valleys. So we said, okay, maybe there's something wrong in what we're doing. So we constructed artificial sequences where we put in known relationships. So we made a sequence in which each base is related to the 25th base. You know, so we set up the first 25 bases, and then the next one we flip a coin, but the coin is biased, four-sided coin, with certain probabilities, so that we introduce some dependencies between the bases. And when we look at the AMI profile, we see a peak at 25. We did this all over the place, so we knew that what we were doing was right. And we get this, and so we start thinking, why is it that we have those peaks and valleys? And we were doing this, and then I remember this day. So I was sitting there looking at it, staring at the data. That's what I do. And said, I wonder if, you know, there are some different kind of structures in chromosome 2. So I ran chromosome 2, and we do it the old-fashioned way. We write a C program, and then we use GNU plot to plot it. So ran the program, got the vector plotted using GNU plot, and it came out with the same plot. And said, okay, I screwed up. I probably plotted the same thing again. Looked at it, I hadn't plotted the same thing again. Then I said, okay, I ran the wrong chromosome through, the same chromosome through. Looked at it, it doesn't seem like it. I downloaded the second chromosome again, ran it again, came with the same thing, said, okay, I did something wrong with the first chromosome. So then I did a diff, which I should have done in the beginning. And there were two different chromosomes, just the way they were supposed to be, but the profiles were the same. And so then I did the next chromosome, and the next chromosome, and the next chromosome, and this is a selection of them. They're all the same, almost. They're not exactly the same. They're all plotted down here in the lowest one. Okay. Remember, all of these chromosomes are of different sizes. They contain different genes. They have different bases. They are different. Okay. You take each chromosome, put it through any program you want. These are different chromosomes. These are different DNA sequences. Yes? Say it again? I'll come to that. Yes, all of them. So here is mouse chromosomes. Right? Here is a worm. Simpler structure, but it's the same, right? And if you look at them side by side, here is human and mouse. Here is the worm and yeast. I remember the day that I saw this, because, you know, there is that moment. Your hair goes up on your hands, and then something cold goes down your spine, and you have this glorious feeling. You saw something, you know? There was all of these bases, and now you saw a pattern. It's there. It was just this beautiful feeling. And that's the thing that you live for, right? I mean, that's what makes all the stupid administrators and their crap, all of it, worth it. All of the times that some reviewer did just dumped your paper because of some, that time, that moment is worth it. I swear to you, I still remember that moment. And there are a few of those moments in your life. They're worth it. It's okay. I'll take the rest of it for that. In fact, you know, I had to take a lot of it for that because it took me 9 years to get this published. I sent it to Nature Genetics, and they split evenly. They had an editorial board meeting, and half of them said this is artifact of the program, whatever program this guy is running, and the other half said this is something useful. And this went on for a while, and then finally they said, we're going to send it to one reviewer, because I kept harassing the editor, so I knew. And that whatever that reviewer says is what we're going to do. So I have had a lot of reviews. This one is my top one, because what he said, and I'm sure it was a he, because there is certain, you know, arrogant stupidity that only males seem to have. So he said, I can tell the difference between a human and a mouse without looking at its chromosome. So what's the point of this? Go away. So we spent a lot of time thinking, okay, what is the point of it? It's really beautiful, I think. I still think it's beautiful. And I still don't understand most of what it's saying. But you know, you end up saying, okay, I'm going to prove something to these guys. I'm going to use this for something. So this was my model. This is the one that I'm going to use from here on. Okay, I'm treating the genomes as a stochastic process. From that stochastic process, I get a model. And that's my model. Okay. And now what I'm going to do is I'm going to try and see if I can make this model useful. Okay, so the first thing you can do is just say, okay, can it distinguish between coding and non-coding regions? And it can. It's not very good. Basically, if you look at this and you look at peaks at multiples of 3, forget about the 3 itself, because that gets overshadowed by the amount of mutual information between 2 and 1. So forget that one, but look at 6 and 9 and 12 and 15 and 18, and you'll see that you have peaks all of those. And that's a coding region. Okay. If you take, here's another coding region which is longer, and therefore we could get better probabilities. Actually, I did this about 4 or 5 days ago, so This was me. But here's a non-coding region, and I screwed up. I'm not very good at this thing, at PowerPoints. But if you look at these, look at the peaks, you'll notice that there are, you know, at 3, well, we're not counting 3, so look at 6. At 6, you actually have a dip. At 15, for example, you can see clearly you have a dip, you don't have a peak. So if you count the peaks, if you just go through and say, is there a peak at 6, is there a peak at 9, is there a peak at 15, and just give a yes/no answer, you get a series of ones or zeros, and then you add up the ones, and that's a very good way of figuring out whether you have a coding region or not. The problem is it has to be a long enough sequence. If it's a short sequence, and there are better ways of doing this, right, of figuring out coding and non-coding regions, even, you know, mathematically, even statistically. So yeah, I went through, of course, I did all of them. But, you know, originally what we came up with, if you remember, we had a profile for the human and a profile for mouse, and they were different. So our argument at the beginning, from the beginning, was that what this is a species-specific model. It tells you what species this DNA sequence comes from. So if it is a species-specific model, then we should be able to, you should be able to use it for phylogeny, for example. That would be one thing that you should be able to use it for. Okay, so here's one particular application. We did a whole bunch of things with all kinds of different. This was a student who made really good PowerPoint slides, so that's why this is the one I'm going to show you. So this is a gold standard phylogeny for yeast, for Candida and Saccharomyces, different species of Candida and Saccharomyces. And this is constructed from 706 orthologous genes. Very, very, very labor intensive. Takes a long time to do this. And basically, this is what you get, right? So instead of taking 706 orthologous genes, what we did was we said, let's take the region between 2 ribosomal genes. So here's the thing. Genes generally tend to be for something. They're used for something. So the mutations in genes is restricted, right? When you mutate a gene, you might mutate its function, which might kill off the organism. So there is a pressure on the genes not to mutate. But if you look at regions between genes, those are free to mutate. You know, unless there's a promoter or something in there, they will generally mutate, and they will be then transferred into the next generation, to the next generation, the next generation, and so on. So they make these internal transcribed regions between genes make good markers for phylogeny. So basically what we did was we took an internally transcribed spacers called ITS region, and then it's about 1,000 bases in this particular case. And we used that to come up with a phylogeny, and this is the way we generated the phylogeny. So we took, we found the AMI profiles for each of these sequences. It takes much, much less than a second to come up with the AMI profile. And then we looked at the distance between the AMI profiles. And we did it 2 different ways, but I'm just going to show you the first one, which is we looked at the code. So think about the AMI profile as a vector, right? So it's, if I take 32 values for the AMI profile, I have a 32-dimensional vector. And I have, this is my AMI profile for this species. This is my AMI profile for the other species. The 2 vectors have an angle between them. And the more similar the species are, if our argument is correct, then the more similar the species are, the more closer these vectors should be. The angle should be less, right? So 1 minus the cosine of that angle should give us an idea of a distance. You know, if the angle is less, the cosine is less. 1 minus the cosine should give us a distance metric. And so that's what we use in there. We just take 1 minus the cosine of the angle, which is simply the dot product of the 2 AMI profiles divided by the magnitudes of the 2 vectors. Okay, really, really, really simple. And then, I mean, I've been railing against software programs, but we used Phylip to generate the tree. Phylip is a good program. And here's what we got. So it's not, it's not as good as the gold standard, but we didn't use 706 orthologous genes. We used just one internal transcribe spacer, and it kept the main clusters. So the Candida, in this case, the Candida are all up in the clade above, and here the clade below is the Candida. Let me see if I can use the pointer. No. So they're together. We have Candida here, we have Saccharomyces up there. If you look at how things are, they're not as many here as there because we couldn't find as many ITS sequences in the database. So databases are useful for something. We didn't, so these were the ones we found. And we've done this in many different things. And this one I thought was interesting because we use only 1,000 bases. If you use more, if you use about 5,000, you get very, very good trees, phylogenetic trees. But the thing is, if it's a model for the species, why should we just use, if I'm going to use ITS region, if I'm going to use the same region for all of them, granted, we don't do any alignment or anything here, but still it's the same region, right? Maybe the other standard methods will do the same. So I used, this was on the internet, phylogeny engine with the ITS spacers, and this is what you get with the standard methods. You still, the outgroup is still there, and are a little bit preserved, but not a whole lot. I mean, Candida dubliensis and Candida albicans, the most important in some ways of the Candida species, are exactly in the same place. You can't differentiate between them. Okay, which is not the case with our approach, right? If you look here, albicans and dupliensis are next to each other, as they should be, but they're separate, right? If you give that same 1,000 bases to this, the standard approach, you don't get anything. Okay, so it's nice for phylogeny, but it's nice for a little bit more than that. I remembered that afterwards. Let me show you. So here we have the same region, the ITS regions, from all of these different species. What if we took different regions from the different species? If it's a model for the species, it shouldn't matter which region we take. The model for the species should be the same everywhere. And I don't have the data here, but it will work. The only thing you have to do is, with 1,000 bases, it doesn't work. You need about 5,000 bases before it works. But if you have 5,000 bases, you get really good differentiation between species, okay, regardless of what the species is, regardless of where you're looking at the differentiation. Okay. We run into problems with bacteria when we try to do this in bacteria because there's a lot of genomic transfer, and because of that, you might randomly end up with the same piece, identical piece in both of them, and that's not going to show any difference. But in general, given a large enough piece, you can do phylogeny without having to worry about which particular gene to pick out. Okay. How am I doing on time? Hey, I better hurry up. We did Go predictions. So this was, again, this is for yeast. We looked at Go terms. So given a particular Go term, we looked at the genes that contain that Go term, and we said that's one class, and then we took the genes that didn't have that Go term, and we said that's a different class, and then we built a classifier using the AMI profiles. Okay, and then what is presented here is the area under the curve for the ROC. It's not bad. right, for certain things. So this is, BP stands for biological process, you know, what the biological process is, where it is, the cellular component, and the molecular function. So molecular function, it does the best. But the left side is for high abundance. So we looked at if that GO term was contained in 10 genes or less, we said it was a low abundance GO term. It was contained in 10 genes or more, we said it was a high abundance GO term. So then we did a classification based on that. So this is just a ROC curve, and this is the AUC, the area under the curve, changing the thresholds. It's okay. It's not that great, but it's okay. You can work with it. Yeah. Here's something else you can do. So this is something where, ignore the things on the axes, but basically what it is I took the the sequences, 100 base pair long sequences, and I used a vector quantizer and ended up with this is a tree-structured vector quantizer, for those of you who are interested in that kind of stuff. And I ended up with a binary label for it. Then I looked at what the distance between the binary labels was. So it's a kind of quantized idea of the distance. Basically, what I was trying to do was say, if I take a sequence, 100 base pairs, and then I move it 50 bases away, or 10 bases away, and I look at the AMI profile now, and if I move it 20 bases away and I look at the AMI profile now, can I tell, are these going to be progressively distanced as far as the AMI profile is concerned as well? And you can see that for up to about 1,000 bases, you have a monotonically increasing distance. Okay? And the way we used this was in sequence assembly. So here was this is all of our algorithm. We took the fragments, shotgun fragments, and then for each of the fragments, we came up with the AMI profile. Then we took those AMI profiles and we clustered them. And I call it vector quantization here. Basically, we clustered them. And then, but remember that in a cluster, you can sometimes get, you know, if you go far enough away, it might look like you're closer. So when we got a cluster, then we did the computationally more intensive thing of we looked within the cluster to see if the clusters kind of look like each other. And that was done just by looking at a very crude alignment of sorts. And if it was, then we broke up the cluster. And then we went through, for each of the clusters then, we did an alignment and put together the sequence. So we get a cluster, so now we align it, now we have a longer contig. And so now we start out, suppose I have 32 clusters, then now I have 32 contigs. Initially you start out with, say, 256 clusters, and you have hundreds of thousands of reads, and you end up with 256 clusters, or maybe because you have to separate them out, you end up with 300 clusters. And then you take each of those clusters and align them, come up with the aligned sequence, and then you do the AMI again, and then you do the clustering again. And now you'll have fewer clusters. And so you keep doing that until the number of clusters doesn't decrease. And that's the number of contigs you have. It's very fast. It's very nice. No de Bruijn graphs involved. No. Nobody uses it, but it's very nice. Maybe someday they'll use it. Okay, so here's another very, very different application. Here, what we did was, remember, we were looking at the DNA sequence, and then we had to calculate the joint probabilities. That's why we needed that long, we needed 5,000 bases. Right? And so the way we calculated it was we counted, okay, how many A's and C's are 6 apart? You know, we went down the string, and that's how we calculated the joint probabilities. In this case, we did it differently. In this case, this is applied to a protein sequence. So these are the amino acids. And here, what we did was we went down. We had a number of, we were looking at clonal populations. So supposedly clonal populations. This was an HIV project. So we were looking at HIV, a whole bunch of HIV sequences from different patients, right? So we looked at, we fixed, in this case, K and M, and then we calculated the probability going down. So these are all aligned. I think I should stop sometime soon. But, and now we have, instead of just having an AMI profile, we have an AMI matrix which has K and M as its two variables. And here is one for, this is for the envelope protein of an HIV-1 virus. It has 303, I think, amino acids. And so that matrix is 303 by 303. And this is kind of what it looks like. Okay, so this was a data set that was kind of spooky. This was a data set from the early 80s. A colleague of mine had been working in Zambia at the time. It's a virologist. And this is before there were any drugs for HIV. And so there was no treatment. So they would get these patients, and he was looking at mothers and infants. So they would get these mothers, and they would have pregnant women who would have babies, and then the babies would either live or die. Okay, and so these sequences that he got, these HIV sequences that he got, were from the babies. And the babies that died, they called those the rapid progressors, and the babies that lived, they called those the slow progressors. So in some cases, the babies lived at least as long as their observation window was. In some cases, they died. So we did this AMI matrix for the different groups, and you can see that they're different, right? So that's the rapid progressor population, and that's the slow progressor population. It's a really, really good model for at least some kinds of problems. Maybe not everything, but some kinds of problems. Okay, I think that's about as much of AMI as I'm going to do. I'm going to tell you a little bit more about metagenomics. So this is another student of mine, former student of mine, whose work this is, so, because I'm running out of time. So here's this problem in metagenomics usually. Right, so we do this a lot. I'm from Nebraska, so we're a very agricultural state. So there's lots of issues with food safety. We're worried about microbiomes all the time. Now, what kind of microbiomes are around? Whether they're good, they're bad, what bacteria are present in the gut, what bacteria are present in the work environment, and whether there's contamination, all kinds of things. So here's the general way in which it happens. You have a number of possible genomes. You don't know what they are. These are in the database. Again, good use of the database. And then you take the microbiome, you get rid of everything but the DNA, and then you eat up the DNA into small fragments, and then from those small fragments, you have to decide what is the abundance of different bacterial species, right? That's the goal. The problem is that a lot of these fragments will get assigned to multiple genomes. And the reason is that a lot of those fragments look like multiple genomes, okay? But this is a very nice place to use communication. So this is just an example of, at that time there were about 1,800 or so bacterial genomes in RefSeq. So we fragmented them and then looked at how similar they are. And you can see the different species. If fragments only belong to one species, you would just see one red line in the middle. But you don't. You see them scattered all over the place. Right? Here's what we do. We say we have a true abundance values. That's X. We don't see those true abundance values. They are the number of different bacteria that actually exist in that microbiome. And then there is what we see. We take those sequences and then we apply any of the different fragment assignment techniques that are available, and there are a ton available. And so we assign them using any one of those, and we get an observed abundance. And the way we formulated it, we said this is a multiple input, multiple output communication system. We have a set of inputs, we have a set of outputs, and there is corruption in the middle. And we don't know what the corruption is, but there is corruption in the middle. So given this kind of a channel, we need to figure out what are the transition probabilities. And we used a very simple metric to come up with the transition probabilities, we looked at what are the multi-reads in different genomes. So how much of two different genomes have in common, how many 100 base pair reads they have in common, and then divide that by the size of the genome itself, and that gives you some idea of a probability, right? Don't go to sleep, it's almost done. And so, then what you can do is, go back. So what you can do now is, you can write up a simple equation. Say P is a matrix consisting of all the probabilities. Then you can write the equation Px equals Y. That's it, Y equals Px. X is the input, P is the transition probability matrix, and Y is the output. Okay. I can observe Y. I figured out a way to kind of generate P, good or bad. And what I have to do is come up with X. Now, if P was well conditioned, I could just say X equals P inverse Y, and I would have the answer, and that would be it. P is very ill conditioned, so you end up using different kind of numerical methods to solve for that equation. But the results are really nice. We called it multiple input, multiple output sequence analysis. Mimosa, everybody has names, so why shouldn't we have a name? And mimosa is a nice drink. It's a nice name. And it does really well. Those were the competitors at the time. There are others that have come out since then. It doesn't matter. Mimosa still works better than anything that's out there, okay, in terms of how well it does assignments. In terms of speed, it does phenomenally well. In terms of memory footprint, it does very, very well. Okay, so, but it's a very simple formulation, and it gives you really nice answers, right? And you know at each step what you're doing. There is no question about what you're doing. And I'll leave you with that. So, you look at something and it's just a jumble of bases. But it isn't just a jumble of bases. There is all kinds of cool things in there. And they're not just, where's the gene and where's not the gene in any given sequence. You have many things going on. You just have to look at it with different eyes. And so my propaganda today is look at it from the point of view of communications. Look at it from the point of view of a stochastic process, and you'll get really nice rewards. I promise. Okay. So here are the bioinformatics people in my lab. I have two parts to my lab. One is the data compression people. They're mostly graduated. But these are all of the bioinformatics people, so it's been a small group, starting from the first. Hassan was a graduate of Boğaz'da, so was Ufuk. Yeah, those two are from Boğaz'da. Yeah, and I'm done. See, you gonna ask any questions? Yes.
I have a lot of questions.
Okay.
Thank you very much for this fantastic talk. Okay. Thank you very much for this fantastic talk. I was wondering if the AMI profile that you detect for the various different organisms is related to chromosome packing.
I don't know. That could be it. That could be a reason, but I don't know enough about histone or histone modification or anything like that to really answer that. I really would like to know, but I don't. Okay. But it could very well be that.
Okay, thank you.
Thank you very much. Did you or somebody else apply these great results to CRISPR technology?
No. No, if you
great
would, that'd be
potential.
Great. That'd be great.
So much. Okay, let's talk. This is very interesting.
Come on, guys, ask. You gotta have some question.
This AMI profile, could that be the result of some optimization or local optimum during the evolution? So
It has to be, right? I mean, if you're going, if it's, we did, so one of the assignments that I give my bioinformatics classes, I give them mitochondrial sequences, and I ask them to come up with a phylogeny for them. And I give them different techniques, and I give them AMI as well, one of the techniques that they have to use. And you can come up with the phylogeny for using the mitochondrial genome for these different, admittedly very different organisms. You know, there are two kinds of whales, but then there's a monkey and there's human, and you know, mostly things that the kids like. They don't like bacteria that much. But yes, so clearly it has something to do with it, but I don't know what. I think my answer is going to be I don't know. Come on, guys, in the back. Come on. Ask something. Just do it. You have to ask something. You were looking very, very, very. You know, you're not going to ask anything. You sure about that? So we had this fantastic. Well, depends. We had this professor in ODTÜ. So that's where I went for my first 3 years. Tomorrow you'll have Kemal Oflazar will give the first talk. And he was in the same class. And one of my very clear memories of, very distinct memories of my undergraduate is waking up in the dorm with Kemal's hands around my neck, promising me that he was going to hurt me really badly. You have to tomorrow, when the question answer period comes, you have to ask him what happened. It's got something to do with the physiology class, but you have to ask him. Okay? Yes? Okay. You're not going to ask any questions? So I was telling you about this professor, Professor Bedri Soer. Everybody hated him. He was the roughest professor I have ever had, and I have no idea why he was that good. But one of the things he used to do was he used to have you there was a class, and he would ask you to come up to the board and solve problems. And there were two theories. One was that if you looked at him directly, he wouldn't ask you, and the other was if you looked away from him, he wouldn't ask you. So half of the class was staring at him, the other half of the class was looking at the floor. I have several people who are just kind of saying, no, don't. You have to ask. If you don't ask, you can't leave. Somebody has to ask a question. Sorunuz var mı? Okay, I'll humiliate myself and speak my really poor Turkish. Bir şey sormanız lazım. Yani öyle oturamazsınız. Come on. I put on a tie. Bunu giydim, kravat giydim ben. Hiç giymiyorum. 2,5 sene oldu. Bunu giyiyorum.
Firstly, I would like to apologize if this is something simple or wrong, because it's not my area of expertise. What you show us at the beginning about the relationship between genes and their distribution, it looked like to me like Fourier distribution. So, can we recreate something like a signal, maybe from them, reverse it?
I think we can, because basically what the AMI does is it kind of looks like an autocorrelation. So you would think that if you took the Fourier transform of that, you would get a power spectral density.
Have you tried to do it? Like, did you be able to get a signal? Maybe like structure?
No, I haven't. I did look at the power spectral densities, but I was having enough problems of trying to get people to accept this. I never really followed it, but I think that's a good idea. And, you know, just looking at it in those terms, looking at it as the autocorrelation function of the signal, and then taking the power spectral density, and then seeing which frequencies are.
I assume this is what you would realize, like those moment of enlightenment. That's what you realize, because That's the light bulb that goes in my head. This looks like a Fourier transform of a signal.
Could very well be. Could very well be. Give it a shot. Figure it out. Yeah?
I will maybe in the future.
It's very trivial. The program to generate the AMI profile is like 20 lines long. That's all it takes. I have my, second year undergraduate students do this for a project. It's that trivial. It really is that trivial. If anybody's interested, I can send you the program. Don't use it. Write your own. But it is that trivial. Yeah? Yes.
Dear Professor, first of all, I would like to thank you for your good presentation. Unfortunately, this is not my expertise area, but I would like to ask a question.
Sure.
I don't know whether it is really important or not, but I'm trying to work on community detection. And I see a figure you showed that us trying to find out grouping genes. Are you using any community detection algorithm or approach, or you are just using a standard k-means or other clustering algorithms?
For the microbial profiling, for the community detection, we're using actually, to solve the equation, we're using a matching pursuit algorithm. So it's a polytope.
Sorry, I couldn't understand. Can you?
Matching pursuits. Because we have a system of equations for which we have an ill-conditioned matrix, right? So we end up with P times X equals Y, and what we want is X. So we can't just take the inverse of P. So what we've tried to do is solve it numerically, and the method that we've used is matching pursuits. There's a paper that came out, I think, in the January issue of IEEE ACM transactions on computational biology. That's this paper. That's Ufuk's paper, basically.
Okay, thank you.
One more. Come on. You're just not going to do it, are you? I mean, think about it. Why? Sormayacak mı? Ha! Aferin sana.
My question is not about biostatistics, but it's about statistics. Beginning of your speech, you said you gave an example of null hypothesis and those examples. Maybe I misunderstood, but you said that when we in null hypothesis, when we prove the alternative hypothesis, even we prove, your example is the probability is 0.00009, I think. Even we prove the alternative hypothesis, do we have to collect the data, or do we need any other evidence? My question.
So the evidence, the problem is that if you got the results, if the steps of the process with which you got the results was clearly defined, like in the case of the coin flipping, the process is clearly defined. You flip the coin and you look heads or tails, right? In that case, you can make a statement that the null hypothesis is true, we accept the null hypothesis, or we reject the null hypothesis, right? That the coin is fair. The problem in bioinformatics is that we have a large number of ill-defined processes. I mean, they're ill-defined in the sense that the person who wrote the programs knows what he did, and for him or her, that process is very well defined. Okay. But for the user, because there are so many parameters that can be adjusted, the process through which the result is obtained is not that well defined. And because of that, you can make claims that are not true, period. And you can mislead yourself, and that's even worse.
Thank you very much.
Teşekkürler.